side role, I'm also a adjunct professor at Queensland University of Technology in Brisbane, Australia. This is where I'm based at the moment. What is tissue cytometry? I'm sure you all are familiar with flow cytometry, which is a technology that has been around for decades. And when you look on PubMed, you will literally find thousands of papers that have used flow cytometry as a tool of research and discovery. It allows to quantify antibody-mediated staining on single cells with one major limitation. These cells are, as the name flow cytometry suggests, in suspension. Tissue and is able to quantify marker expression on single cells, but in tissue context. We can also identify multicellular structures in tissue and determine tumor areas, the tumor microenvironment, and do what is called spatial immunophenotyping. We can also look inside single cells and analyze subcellular compartments and also quantify pathogens like bacteria or the viral load on single cells or intracellular parasites. Why is quantification important? Well, microscopy has been around for literally a century. It has been a vital technique in biomedical research for major discoveries that have been made, but most of the time it is purely visual. And this leads me to a survey that has been published a few years ago by Nature, a survey done among 1,500 scientists, which stated that 70% of researchers have difficulties in reproducing published data, and additional 20% have even problems in reproducing their own data, which raises the question whether there is a reproducibility crisis in our science. So a call has been raised for a better mentorship. This is something the universities have to take care of. The proper use of statistics, which is something each PI and each lab has to make sure they use the proper tools, and more robust and stable experimental designs. This is something where TissueGnostics can come in. Who is TissueGnostics? We are a medtech company with our headquarters in the city of Vienna in Austria, in Europe, but we have a structure of subsidiaries and offices practically on all continents. By end of last year, our list of reference papers exceeded the number 1,950. So this month we will actually reach 2,000 from research groups in more than 60 countries on all continents. I would like to emphasize that TissueGnostics is not only a software company, we are a manufacturer of medical devices, and the company has been certified for ISO 13485, which is the main quality norm for medical products, already back in 2008. I myself have been trained as a cell biologist, and the company has its source in academia. So we have still strong ties and close links to academic research, and almost 24 of nationally Austrian public funded research projects have been conducted so far. The latest one is of Vienna, where we work on advanced deep learning solutions for single cell recognition. I would also like to mention the 5 biggest research consortia which we have joined over the past 10 years. All 5 of those are EU-funded multinational Marie Curie innovative training networks. These were Alcatraz, which deals with the anaplastic lymphoma kinase, headed by University of Cambridge in UK. 8path for next generation digital pathology, headed by University of Nottingham in UK. The calcium sensing receptor network, headed by Medical University of Vienna. The European network on cell migration studies, headed by Technical University in Aachen. Those 4 are already finished. Currently ongoing is Helical, big data analysis for clinical benefit by machine learning. This is headed by Trinity College Dublin. TissueGnostics has a set of instruments for slide scanning and subsequent image analysis. Field scanner for histological slides, which looks like this. Our main product line is called TissueFAXS. This is based on a high-end microscope of Carl Zeiss. So TissueGnostics is OEM partner of Carl Zeiss Germany. The instrument is available in different configurations for bright field only, fluorescence only, or fluorescence and bright field scanning. There is an inverted version where instead of 8 slides, you can also put a micro well plate from 6 well to 384 wells. Then there is a high throughput version with a slide loader for 120 slides, and all fluorescent configurations can be equipped with a spinning disc confocal for whole slide confocal imaging. Like with any slide scanner on the market, the immediate result of the TissueFAXS scanning process is a virtual slide. We see here a prostate section in a 4-color staining. In orange is the tumor marker. In red are the secretory epithelial cells. In green, the basal epithelial cells in the prostate. And we can literally see how the tumor invades here the normal adjacent tissue. We can, like with Google Earth, navigate through the sample and zoom in to see the detail down to the subcellular details, like here structures inside the nucleus and in the cytoplasm of the secretory epithelial cells. It also works with cell cultures. Here we see a neuronal cell culture in a 5-color staining, where we can also navigate through the virtual slide and zoom in to see this, the details. You can scan all kinds of samples, paraffin embedded, cryocuts, cryocut sections, biopsies. There's a special workflow for TMA, tissue microarrays, and so on. I would also like to mention that we can scan oversized slides. So, you know, double-sized, quadruple slides, or even slides which have the size of 8 single slides. So glass plates filling the entire stage. There's also a possibility to combine the inverted system with a culture chamber and do live cell imaging and time-lapse microscopy. To give you an idea about the performance on the confocal version of TissueFAXS, we see here a whole mouse brain section. This is courtesy of Howard Hughes Medical Institute, the Janelia Research Campus, Janelia Farm Research Campus in Ashburn. In order to scan the entire mouse section, 350 individual fields of view had to be acquired. Each field of view here, the rectangle, what you see is one high power field of view at 20x. Each field of view consists of 4 channels. Each channel has 21 layers. So in total, 29,400 images or 115 gigabytes of compressed data. Scanning in a single layer takes of the whole mouse brain section takes 8.5 minutes. Of the entire set stack with 29,400 images, 1.5 hours, which is still very fast. If you try to do this manually, first, it will not be possible to do it manually, but if you try, you will probably sit a week. Here, it's done in 1.5 hours. But all these images, as beautiful as they may be, are just an intermediary result. What makes TissueGnostics is the image cytometry software, or actually there are 3 applications that we offer. They are called HistoQuest, which is single cell analysis for bright field samples, or immunohistochemistry, TissueQuest, single cell analysis for fluorescence, and StrataQuest, which is our high-end solution that can do everything what HistoQuest and TissueQuest can do, plus much more, like contextual tissue cytometry, distance measurements. There are deep learning algorithms and extensive machine learning module, which I will show a little bit later. As a work core idea in the whole TissueFAXS workflow is that we use the nuclear stain, hematoxylin in bright field, or DAPI in fluorescence, as a so-called master channel to identify the individual nuclei. Here you see a standard peroxidase staining of a colon. In brown is a well-known marker, Ki67, so proliferation marker. And the obvious question is how many nuclei are proliferating, or in other words, how many nuclei are stained in blue. We have shown exactly this image to researchers, to more than 200 researchers, and asked them, what do you think, how many cells are positive in percent? When we say everything we see is 100%. And believe it or not, the estimations from experts ranged from 1 to 40%, which I would say is a substantial difference. But this shows the main problem associated with visual evaluation. You always get a range of estimations rather than a reproducible measurement. With the TissueFAXS platform, we use, as I said, the nuclear marker as master channel, and the yellow contours here show you what the individual nuclei, what the algorithm has identified as individual nuclei, and then measures for each nucleus how much blue and how much brown is associated with it. And this is shown in such a scattergram as you know it from flow cytometry. Just that now we have hematoxylin on the blue, on the x-axis in blue, and Ki67 brown on the y-axis. Here is this famous line known from flow cytometry, the cutoff, which has to be set in accordance to an isotype matched negative control and determines the specificity of our staining. Only cells with a brown intensity greater than 20 here will be considered as positive. Everything else below 20 will be considered as Ki67 negative. And as we see, this is the result, 14.5%. And the important point is, whenever we do this analysis on Monday morning, on Wednesday noon, on Friday afternoon, in Singapore, in Brisbane, in Vienna, we will always get the same result, a reproducible measurement, rather than a more or less arbitrary number based on estimation. Ki67 is an example of a nuclear marker that we just saw. So here we use the master mask, the marker of interest. In case our molecules are expressed in the cytoplasm, we use a slightly modified approach. We still take the nuclear stain as master channel, but then generate a secondary measure mask around each nucleus to measure cytoplasmic reactivity, as we can see it here. This is from a skin cancer research project. The marker is named COP1, but that's irrelevant. It's about the methodology. We can see here, this is a biopsy taken in normal skin, that this marker is constitutively expressed in the epidermis. After UV irradiation, however, the expression of the marker increases in two ways. The amount per cell increases, but also the number of cells which express the marker increases. So there are more cells are positive, and this is our 2+ positive, or your colleague then maybe says, well, I would say it's 3+ positive. Rather than discussing how many pluses you assign, TissueFAXS will give you an observer-independent measurement. We have here the skin sample before without UV irradiation, and we see that there's 16.8% of the cells that are positive, up to a mean relative fluorescence of 90. After UV irradiation, this increases to 44.8, and we can see there is a shift towards higher values up to 130, which reflects what we visually perceive as brighter. But again, this is a measurement here. This insert, the so-called quadrant statistic, shows us how many events are where in the scattergram, because these two cutoffs, this is the cutoff on the x-axis, this is the cutoff from the y-axis, separate our diagram into 4 quadrants: the upper left, the upper right, the lower left, and the lower right. And this quadrant statistic tells us how many cells are where in total events, in cells per square millimeter of tissue, and in percentage. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells between virtual slide and data space. So you can always click on any cell or other object that has been identified, like glands or multicellular structures, which we will see later. You can click on the cell and it will start blinking in the scattergram. So you know exactly where this cell is located in data space. Following our motto from image to data, we consider this a step forward, and thus this has been named the forward connection. It also works the other way around, back from data space to the image. Like in flow cytometry, you can make a gate, a selection of a subpopulation of cells here shown in orange. Also in other scattergrams, the orange or whatever color it is will appear. So you see how the population is distributed when you look on different marker or parameter combinations. But you can also visualize in the virtual slide where are the cells. So those cells in red here are from inside this gate. That means you have an immediate link between cellular phenotype and histology. Where do certain cells occur in the tissue section? And this has been called the backward connection or reverse gating. A very important application in the field of immunology is that we can use this platform TissueFAXS to determine the immune status in situ. With flow cytometry, we can determine the immune status in blood, so systemically. Here we can do it in specific tissue. We see here an example of a 4-color staining with DAPI in blue, T helper cells in yellow, cytotoxic T cells in red, and FOXP3 in green. You may know that FOXP3, CD4 double positive cells are considered the regulatory T cells. And this is actually what this is about here. This is from a research group at University of Western Australia in Perth. We use the nuclear marker indicated here with the green contours as a master channel, and then measure how much yellow, how much red, and how much green is there. Here we see the backward connection on CD4 with the red contours, here on CD8, and finally the co-expression of CD4 and FOXP3. So this is in the upper right quadrant are the double positive, in the lower left quadrant the double negative, in the upper left and the lower right are the single positive. And each, of course, quadrant has its own statistics, gives you a percentage, number, and density value. There are many applications among those 2,000 reference papers that we have. There are also clinical studies that have been performed with our platform, like here for Novartis USA, a clinical study on an agent called pimecrolimus. This is the Elidel, the active agent in the Elidel cream to treat atopic dermatitis. For pharmaceutical companies, of course, it's important to work with a validated technology and determine the inflammatory status in situ. So it's not enough when a dermatologist looks at the skin and says, well, less red than last time. But with this in situ approach, it was possible to really quantify how the number of inflammatory cells of each the different immunological protocols that you can see here were applied. This is just CD45, CD14 as one example. All this is based on negative controls, as I mentioned earlier. But the key figure is here on the lower right. We see here the individual phenotypes. In black are those atopic dermatitis patients that received the active agent, pimecrolimus, and in white, the control group. And we see that the patients which received the actual cream, the Elidel cream with the active agent pimecrolimus, showed a significant decrease in the number of inflammatory cells compared to the control group. So this was a functional proof that this cream works in situ, and as I said, not just a guess or a rough estimation. We have now seen an example of single cell analysis. We can also look into the cells, so in subcellular compartments. Here we have an example from a client in Sao Paulo, University of Sao Paulo in Brazil, detection of activated microglia. We detect the cell bodies in yellow and the filaments here in green. And this scattergram shows us now 3 groups of filaments. In orange now, which is also the active backward connection here, we see the distant filaments. Now the dots here do not represent individual cells, but single filaments. And these are all those filaments which are more than 50 micrometer away from the next cell body. In red are the short and attached filaments. Short means shorter than 40 micrometer in length and attached to a cell body. And in blue are the filaments which are attached to cell body but longer than 40 or 50 micrometer in length. So and this is mouse brain. So in this study, they analyzed mice which were treated with different agents in different ways. And by this analysis, they could determine the degree of fragmentation of the microglia. Another example of subcellular analysis is the determination of intracellular parasites. Here using leishmaniasis as an example. This was a collaborative project with the Institute of Immunology, Uwe Ritter's lab in Regensburg in Germany. I don't have the time to go into the details of the disease, but very briefly, leishmaniasis is parasites, intracellular parasites that are transmitted by mosquitoes. They infect cells like macrophages, and patients suffering from this disease really see severe structural tissue damage. In Regensburg, they were looking on ways to kill the parasites, but leaving the human cells alive. And they worked in human macrophage cultures on the inverted system in microwell plates, as we can see it here. This is one virtual well. The individual rectangles that you see here are the individual fields of view that had to be acquired in order to capture a full well from a 24 well plate. Here we see the DAPI stain, and when we look in a bit further, the small dots here, these are the parasites. So first we identified the nuclei of the macrophages here, then generated what is called a ring mask. You can see here, each of the nuclei has a ring around it. And then we looked inside the ring for the small dots. So it's the same staining, DAPI, but distinguished by the size of the structure, because macrophages have a relatively big nucleus compared to the intracellular parasites, which have a small nucleus. And the yellow dots here show you the result of the nuclear detection for the parasites. The algorithm, like any algorithm, does of course not really understand what is a parasite. You know, it analyzes pixels and groups of pixels, intensities, pattern, or whatever. So it's important that you work with culture without parasites. And what we see here, this would be false positive. Here the algorithm detected just background structures, pixels, random pixels, which were too bright. So you set the sensitivity on such actual sample here with Leishmania major. And what it finds here, these are the real parasites. And this is what was also published, and there was also a comparison of automated detection with manual counting, showing a very good correlation. Now, on my first slide, I also mentioned that TissueGnostics is working under the in the scope of precision medicine. It's very important these days, and in particular in precision medicine, to increase the number of molecular markers that we can analyze and quantify simultaneously at the same time on the same sample. So not just consecutive sections, but on the same sections on the same cell type. There are two main approaches to do this. The first one is called multiplexing, where you restain the same sample, always using the same combination of 3-4 fluorochromes, but in each staining round, the fluorochromes are linked with different or represent different markers. And this an example of such a multiplexing approach has been published in 2019 by a group from Yale University in cytometry, and they're using our TissueFAXS system and then subsequently StrataQuest software in a circular approach where they restain, rescan, and then in StrataQuest combine the channels using the cross-correlation algorithm so that in the end you have a virtual 10, 12, 15, 20 color experiment. And here you can see the readout. Please note that each dot that you see represents a single cell in a tissue section, and all plots with the different markers shown here represent the same sample. So this is really a co-expression analysis and an in situ analysis on more than 10 markers. A second, a technical approach to increase the number of markers that can be determined simultaneously on the same sample is multispectral imaging. In multiplexing, we restain with a and then scan with a standard microscope configuration. In multispectral imaging, we use a special configuration, and that is a spectral camera with a liquid crystal tunable filter. This is on our upright system for 8 slides, but also available in a high throughput version with a slide loader. And this allows to detect a lambda stack. What is a lambda stack? Similar to the Z stack or C stack that you know from confocal microscopes. In the C stack, you look at the given region or sample in different focal positions. In a lambda stack or lambda cube, you look at the sample in the same focus position, but in different spectral intervals. And then use the reference, the emission spectra of the individual fluorochromes as reference, and the computer can determine how much from the total emission originates from fluorochrome 1, 2, 3, 4, 5, 6, 7, 8, and so on. Here we see an example of a mouse spleen. On the left-hand side is the raw data set. Looks a bit ugly. Overexposed, unspecific, the misdetector crosstalk. But after unmixing here on the right-hand side, we can see the individual zones of the spleen and the individual markers here, like smooth muscle cells, endothelial cells, monocytes, T cells very clearly, yeah? And based on specific staining. A prominent paper in which this TissueFAXS SPECTRA technology was used was published in August 2020, where the immune response against the SARS-CoV-2 virus was quantified in situ in the lung tissue of COVID-19 patients. And this was probably the first paper that showed that we have a significant problem with herd immunity. If you recall, in early 2020, after the onset of the pandemic, there were some countries that said, oh, we don't do anything, we wait for herd immunity to come. And this study already indicated in summer, also northern summer, August 2020, that this will not work. This concept will fail, and we know now that it does fail. But methodically, this in situ analysis was determined or was conducted by using TissueFAXS SPECTRA and StrataQuest. Now, in StrataQuest, we have extensive solutions for machine learning and also an advanced algorithm for deep learning, an advanced deep learning algorithm for nuclear segmentation, which is specifically qualified to recognize individual cells in high density environments, which we typically see in lymphatic tissue. This is a classical nuclear segmentation algorithm, which works pretty well here where the cell density is not too high, but when it comes to really dense tissue conditions, the algorithm has problems. And here you see the result of the DNN, the deep neuronal network-based nuclear segmentation, which brings a great improvement in single cell detection. And here's the direct comparison between the deep learning based and the classical image analysis based algorithm. In the machine learning module, we can quickly train a classifier. There are different technologies like support vector machines, other boost, decision trees, random forest classifiers, and so on. You highlight what you're looking for and then run the training, which typically takes a few minutes, and then you can apply the training set to the entire sample. And let me point out that this is not limited, like with some other software, to 2 or 3 classes. As we can see here in this lymphatic tissue, we have 9 classes, including the background, 8 real tissue classes identified, and it still works very well and can be then applied to the entire tissue section and to do the classification. And then you can combine this with the single cell recognition, and you get the readout how many cells, and if there's a proper staining like leukocyte subtypes, how many cells of which subtypes are in which zone of your tissue, and the zones being recognized by machine learning and classified automatically. We have seen that we can look on single cells, on subcellular compartments, on multicellular structures. We can do this in bright field, in wide field fluorescence, in confocal, in multiplexing, in multispectral imaging. Now we can combine all this and perform what we call contextual image or tissue cytometry. We see here a sample of a colorectal cancer stained with 5 markers. Dark blue is DAPI, light blue is cytokeratin, red is CD4, green is FOXP3, and white, some events here, is Ki67. And the question is here, how many T helper cells, CD4 positive T cells, are present, how many are inside the tumor, how many are in the tumor stroma, and if they are in the stroma, how far are they away from the next tumor area? So we identify the individual nuclei. As we have seen this and discussed this before, we use the marker cytokeratin to identify the tumor area here indicated by this orange mask, and then can combine these 2 masks and ask the relevant questions. How many CD4 positive cells are there and where are they? We can come up with such a scatter plot. This is what we call an input gate on CD4. That means each dot that you see here represents a CD4 positive T helper cell. All cells have been analyzed, but in this scattergram, only the CD4 positive cells are shown. On the x-axis is the size of the nucleus for each of the cells, and on the y-axis is the distance in micrometer of each respective nucleus, of each respective lymphocyte from the next tumor area. So we can generate here such gates, which represent distance zones and contain all the CD4 positive cells up to 25 micrometer away from the tumor, 25 to 50, 50 to 100, and so on. And we can use this reverse gating or backward connection feature I showed earlier. And then we see here in red, as orange is the tumor area, and in red are all those CD4 positive T lymphocytes, which are just outside the tumor, but not further away than 25 micrometer. And with this we can do, obviously, for all the gates, for all the distance zones that we have created. And if we go back and then look into this quadrant statistic, we see here, there is a total of 7,234 CD4 positive cells, 2,267 or 31.3% are in the distance zone 1 to 25 micrometer, or here we have 1593 or 22 in the distance zone 25 to 50, and this goes further down here. And we see there's a steep decline. So we have a gradient towards the tumor, or actually away from the tumor. It's highest close to the tumor, and then falls the number of infiltrating cells declines significantly. So we have a very detailed in situ analysis on the single cell level in spatial context. I have another example, which is called spatial immunophenotyping. This is a multispectral imaging using immune checkpoint staining. Here is the unmixed sample, and this would be the individual markers that have been applied here. And here you see the result. But what I want to show here is that you can use what in flow cytometry is called gate arithmetic. So you can make logical gates. Let's look on here, this gate. It shows as this scattergram. It shows PDL1 size. So basically, this is the size of the cells, and of all cells, in fact. Yeah. And here is the PDL1 mean intensity. We make a gate in green on the PDL1 positive cells and use this as input gate for the second scattergram. That means all dots that we see in the second scattergram come from this gate. PDL1 negative cells are no more visible here. Then we look on this lower left quadrant here, where the CD4 negative and PD1 negative cells are, and use this as input gate in our third scattergram. So here we see only cells which are CD4 negative, PD1 negative, and because here we have an input gate on PDL1 positive, we know those cells are also PDL1 positive. And here we have CD8 versus CD68. And when we look again at the double negative cells here and make the backward connection, you see the red contours in the image, then we know here we are looking on PDL1 positive, CD4 negative, PD1 negative, CD8 negative, CD68 negative cells. And of course, you can do this for each combination and each quadrant, and you get a detailed statistical analysis, but also a visualization in the virtual slide. You have a link between data space and the original image. Let me remind you, all this has been done by the TissueFAXS platform in one of our configurations that are available. And there are a number of publications in which the reproducibility of this technology has been tested. One was published by myself many years ago, where we used peripheral blood leukocytes and analyzed them on a standard flow cytometer, and then on our instrument. So the technology was called microscopy-based multicolor tissue cytometry, MMTC, but the instrument is called TissueFAXS, and these are peripheral blood leukocytes. After analyzing on FACS, we dropped on a slide cover slip, and you see here that there's a very good correlation in the analysis, in the measurement or in the result of the populations. Then the cell populations that were determined on FACS and on TissueFAXS. Here, actually a user from Perth has made a comparison between our software, Strata Quest, FOXP3 positive cells were counted, and 3 observers, 3 human experts visually, manually counted the cells. The first thing that we see is that each observer had a different count. That's always the problem with the so-called gold standard with visual evaluation. So a median was calculated or computed from the 3 observers, and then this was compared to the software result, showing a very good correlation all over. Now, I hope with this I could give you first and introduction to TissueFAXS cytometry platform and show you that this supports researchers to move from image to data and thereby also helps to increase the observer independence and reproducibility. Just a few more slides to introduce you to my work at QUT, Queensland University of Technology in Brisbane, where I'm currently working in a group on the genetic complexity of prostate cancer. And I myself am building a working group that focuses on AI-based decision support systems for precision cancer diagnostics. And the project that we have started here is called the Virtual Histopathologist for Precision Cancer Diagnostics. It consists of 5 sub-projects. The first one, because we use prostate cancer as a disease model in the beginning, the platform is not meant for prostate cancer only, but we need to start with something, and this was prostate cancer. In the sub-project 1, we define the markers which are relevant. This is partially a literature study, or anyhow known. In sub-project 2, this is the wet lab work. We develop antibodies, reclone them with a CTIT linker technology, which allows to reversibly link and get rid of fluorochromes. So the idea is to have here multiplexing staining kits, where you stain like 5 markers, but then the whole sample, whole slide imaging, you do not need to bleach, which would take forever, but you can just kind of switch off, open the linker and wash away the fluorochromes. Sub-project 3 focuses on the integration of genetic information to the functional protein level. Sub-project 4, that's the core part for the AI work, where we develop the platform for ISO 13485 compliant image analysis using machine learning technologies, but also data mining, because when I said multiplexing, so we may end up to have dozens of markers. And if you have ever looked at a 4-color or 5-color immunofluorescence, you know how difficult it is to really understand what the 4 colors show us. And now imagine you have 20, 30 or more markers that you have to comprehend how they are connected and interrelated with each other. It's not possible. So we have here, this is a so-called chord diagram, which shows the relationships between the individual markers in form of these lines. The color and the thickness of the lines indicates which relationship exists between each marker to each other marker that analyzed. And sub-project 5, that's kind of the academic base work, where we look in what happens actually in such a deep learning neuronal network. Because as a matter of fact, especially in deep learning, we do not really understand why we get a certain result. We define the input, the images that we give, and we see what comes out. But what happens, or why the outcome is poorly to not at all understood. So we try to bring some light in this black box of deep learning. So if you are interested to learn more or even to join the project, I invite you to contact me. This is my email address at QUT. This is my email address at TissueGnostics. Either one is fine. And finally, I invite you to have a look at our website on the research section where the reference papers are published. As I said, this month we will reach 2,000, but by end of 21, we had 1,950 papers published in 789 different journals from research groups in more than 60 countries. And here you see how the publications per year increase from the foundation of the company to last year, also the average impact factor per product. And finally, I would like to point your attention to this review article, which was published from my group last year in Genes, where we also look on next generation digital histopathology with focus on the tumor microenvironment. So I would like to acknowledge our collaborators from the Marie Curie EU-funded networks and here in Brisbane, and our global partners from the Virtual Histopathology project, which come from multiple continents, also to make sure we consider the genetic heterogeneity of prostate cancer and the different phenotypes that may come based on this heterogeneity. Just last month, we have published a dossier in Austria looking on artificial intelligence in biomedicine. It's not really a scientific paper. It's what funding agencies consider an outreach, public outreach activity. But I think it's still very nice because many of our partners and clients around the world have contributed. So if you would like to have a PDF copy of it, feel free to contact me as well. In this sense, I would like to conclude my presentation. Thank you very much for your attention, and I'll be, of course, happy to take any questions. Thank you very much.
Thank you, Dr. Rupert, for an excellent presentation to introduce the TissueFAXS tissue cytometry. I believe this technology will bring a giant step in computational pathology, cellular image analysis, and even future precision medicine. I think Dr. Rupert is ready for questions. You can drop them in the chat box, or you just open your microphone is also okay. So here I received some questions from Dr. Arnab. Dr. Arnab mentioned this is an excellent presentation. Congratulations for coming up with the idea and tool. The question is, can we build our own algorithm based on the expression of certain markers and their interplay? Will we be able to hold this IP individually for that algorithm?
So in the StrataQuest platform, you can indeed build your own algorithm. StrataQuest is a combination of a general purpose image analysis environment with specific proprietary algorithms from TissueGnostics. So you can combine them to build your own image analysis solutions. And by the way, they are not limited to biomedical research. Just as an example, the BMW Research Center in Munich had a project where they used StrataQuest as well, where they made microscopic section of motor blocks for future car engine and analyzed them. Yeah, so StrataQuest can handle any kind of image, or even X-ray or ultrasound or satellite images theoretically, and you can analyze them, build your own algorithms. There's an interface in StrataQuest to dock or kind of plug in algorithms from MATLAB, ImageJ, Python scripts, or even C++ code if you have. And so you can develop whatever you would like to develop. The algorithms from TissueGnostics and StrataQuest, of course, are proprietary to TissueGnostics. But when you use this tool to develop your own algorithm, then this is your algorithm. You do not need to disclose to TissueGnostics what you do or how you do. And of course, you can also patent this algorithm. Of course, StrataQuest is owned by TissueGnostics, and the algorithm, which are proprietary to us, will belong to us. But when you build on them and make your own algorithm, that is your algorithm.
Mm-hmm. Okay, thank you. Okay, so another question is, Ki67 is a proliferation marker and is often used. It is possible to separate the Ki67 expression in each cell based on their cell cycle stage, as we do in flow cytometry by co-standing with DAPI.
This is a difficult question in that how do you determine the cell cycle? This is very difficult in tissue sections because, you know, excuse me, in tissue sections, you have a cut. Most of the nuclei are cut, yeah? And it's hard to use the normal DNA content measurement to determine the stage of the cell cycle. And actually, hematoxylin staining, as we know, is not quantitative. So in bright field, this anyhow does not work. We have users who do this, who determine the cell cycle stage like G0, G1, S and M phase, G2 phase in cell cultures. Because in a cell culture, you have the complete cell, the complete nucleus, and there you can do it. But in a tissue section, it's very difficult. So you might rather need to apply some cell cycle specific markers like cyclins or cyclin dependent kinases to determine the cell cycle phase rather than using the DNA content as such, which is not really feasible in a cut tissue section.
Okay, great. We can do from the culture cell. Great. Yep. So if anyone have questions, you can drop on in the chat box or just turn on your microphone. Okay.
So Rupert, Rupert,
sure.
this is Arnav again. Thanks for the wonderful presentation and the excellent answers. Another question, at the end of your slide, you shared something on the 7-color spatial phenotyping, almost at the end. So to do something like that, what would be the ideal instrument?
So you're referring to this slide?
Yes.
Okay.
To do something like that, what should be the ideal instrument we would be using?
So this particular example was done with TissueFax Spectra, the version with the spectral camera with liquid crystal tunable filter, where in this case it's 7, 2, 4, 6, 7, but up to 8 markers can be acquired and spectrally separated in a simultaneous staining on the same sample. We have in fact a client in Boston, he has established a 10-color staining protocol based on TissueFax Spectra.
Okay.
These data come from the spectral imaging system.
So this is exactly what we would like to do. I mean, what we are interested in. So I already had some other discussions with your team and also Giridhar. So once we can follow it up later offline. Thank you.
Thank you. Thank you very much.
Thank you. Does anyone have other questions? Okay. If no questions, we thanks Dr. Rupert provide this very excellent, fantastic presentation. And thanks your time to join this webinar. We record this webinar. Please contact us if you need this video. Okay?
Yeah, I would love to have this video and share it with my team so everyone can appreciate the fantastic work that's been going on.
Yep.
Thank you very much.
Thank you.
Much appreciated. My pleasure.
Thanks, Rupert. Look forward to meet to you if you ever come to India. Just let me know.
It's on my to-do list, definitely.
Okay, wonderful.
Thank you very much.
We'll catch up. Thank you.
Okay,
bye.
Thank you. Thank you, everyone. Bye-bye. Thank you. Bye-bye.